which was the other alternative. And then he swallowed a piece of bezoar stone, and it didn't work. And the cook succumbed to the poison. Why do I tell this story? Because this likely was the first ever clinical trial in history. Because it was a real experiment. They wanted to check whether or not the substance, which worked as a potentially as a drug, did it have any effect? Well, it turned out that it didn't work. So this clinical trial turned out to be negative, like most clinical trial actually turned out to be. But Ambroise Paré was a very interesting character. Now, in those days, of course, physicians really learned by apprenticing. There were no medical schools. You learned by following someone who had followed someone else, etc. You became a physician. And in those days, on the battlefield, if you got a wound from a sword or from a bullet, which was bleeding, what they would do was to cauterize it with hot oil, which is what the guy is doing here. But being treated with hot oil was often more deadly than the original wound. And many of these patients died of shock. Well, this is when Ambroise Paré came to the fore with this very interesting story. He, instead of using hot oil, because on one occasion it wasn't available, so he tried to use just some plant extract and some turpentine that he put on the wound. And it turned out that the person survived. Well, this was an interesting finding. And again, it was sort of a clinical trial because he repeated it and found that using these natural products instead of the cauterization was better. And he had one other contribution, Ambroise Paré. You know, in those days, they would amputate very readily. If you had an infection, you know, you had something oozing out of a wound, the only way to survive was amputation. But he found that, you know, upon amputation, very often the person bled to death or died from the heat of the cauterization. He came up with a surgical technique of, instead of cauterization, of actually suturing the arteries shut, much like they do today. But this was in the 16th century. So it's very interesting to back that, you know, they were actually doing these kind of trials back then. But more famous was James Lind. James Lind was an English physician who knew all about a terrible disease called scurvy. And this afflicted a lot of sailors because in those days, ocean voyages were very long, took weeks to cross the ocean. What did they have for food? They had salted meat and crackers. This was before the age of canning, obviously before refrigeration. So no question that there would be an epidemic of scurvy because it didn't have any fruits or vegetables. Today, of course, we know that scurvy is a deficiency of vitamin C. They didn't know that. And the symptoms of scurvy are terrible. Your gums begin to recede, you get these sores, and eventually you die. But James Lind carried out a clinical trial, a very simple one. He took some of the sailors on a British sailing vessel, and he organized a pretty interesting trial. He selected 6 pairs of men to whom he gave either a daily dose of cider, dilute acid, vinegar, seawater, a mishmash of garlic, mustard seed, radish root, or 2 oranges and a lemon. I mean, basically everything you could think of, right? And it turned out that the guys who got the lime juice or the orange juice survived. And this is when the treatment of scurvy using citrus fruits began. It came out of this rather simple and primitive clinical trial, and he wrote this up. Here is the amazing little booklet, a treatise on the scurvy, where for the first time he documented the treatment of the disease with citrus fruits. It didn't take right away because, of course, this was before the internet, so the information didn't get around that quickly. And it took about 30 years until the British Navy instituted the dispensing of lime juice to sailors. And that's why today Brits are sometimes called limeys because of the drinking of the lime juice. And this is a tradition that continued until quite recently. Symbolically, of course, but sailors on British ships were given lime juice on a daily basis, even though, of course, they no longer had to worry about scurvy. But what is interesting about this story is that Lind was the first person to publish this information based on the little trial that he did. Whereas actually, Jacques Cartier, who came to Quebec earlier, centuries earlier, also documented his men suffering from scurvy. Of course, it wasn't named that. Legs became swollen and puffed up, while the sinews contracted and turned coal black, and in some cases all blotched with drops of purplish blood, etc. And Jacques Cartier got some advice from the natives who were living in Quebec at the time. They also knew about this disease, and they had found through trial and error that drinking spruce beer, that is something that was brewed from the spruce leaves, countered this disease. Well, it turns out that it does contain some vitamin C. But Jacques Cartier never documented this properly, never wrote it up in any kind of medical journal. But there's no question that he was, aware of a treatment for scurvy long before Lind. William Withering in England was a country physician who treated a lot of people who had heart problems. And he heard some anecdotes about treatment of this disease by what he called an old wife. This indeed was the origin of the expression old wives' tale. That old wife was a woman who was referred to as Mother Hutton, who was treating people with an extract, a tea, made from the flowers of the foxglove plant. And Withering listened to this story, and he didn't have any real medicine for his patients, and he decided to try it. And he found that it worked, that when people had what we would now recognize as congestive heart failure, where you have swollen ankles and swollen hands, symptoms that Trump seems to have, feel so sorry for him. Withering wrote it up, and there is his account of the foxglove, because he actually carried out a small clinical trial where he gave foxglove extract to some of his patients and not to others. Today, interestingly enough, he is remembered in many ways. Besides the document that he published, there's a pub, the William Withering pub, where it's not obviously foxglove extract that is consumed. But today, the story is fascinating because we know that there actually is an active ingredient in the foxglove, which is digoxin, or digitalis was the extract as it was known. And today, congestive heart disease is still treated with digoxin. So it all goes back to that rather interesting story of William Withering, the old wives' tale, upon which he capitalized, and he actually carried out a clinical trial. So what we really want to discuss here today, though, is how a new drug gets to the market, right? I mean, in those days, there really were not organized clinical trials. I mean, there were these smatterings of efforts by Lind and Withering. Today, of course, the story is very different. So let's explore how a new drug gets to the market. Well, the first thing is you have to have an idea, right? You have to have some idea of where you want to look, what is possible in terms of a new medication. Where can you look? Well, of course, you can look first through history. And that is exactly what we were talking about with James Lind and William Withering. You know, they happened to make these discoveries and eventually turned into real pharmaceutical products. So there are other such interesting medications that came out of from scrutinizing history. For example, there are all kinds of historical accounts about the use of the mandrake root. And it was used as a painkiller and used as a sedative. They never knew, of course, what it contained in terms of any potential active ingredient, but they just knew that it worked. And today we understand why. Scopolamine and atropine, scopolamine you may have heard of because it is used for motion sickness. Atropine is used by ophthalmologists to dilate the pupil of the eye. It's also used to speed up an ailing heart. When someone suffers from bradycardia, that is a slow heartbeat. So the clue was that ancient people used the mandrake root. And modern science now has discovered that there actually are active ingredients in there. So that finding came out of these historical accounts. Another one is the historical account of the bark of the willow tree. Now, in many books, many texts, they refer back to Hippocrates, the father of medicine, as he is called, as being the first person to recommend chewing the bark of the willow tree for pain. The fact is that's a myth. There's no historical evidence that Hippocrates ever did that, and yet it is in textbooks. What we do know, however, is that much later than Hippocrates, an English physician by the name of Edward Stone discovered that chewing the bark of the willow tree did alleviate pain and did reduce fever. Why? Because it contains a compound called salicin. And salicin is a fever-reducing, painkiller, anti-inflammatory substance. In fact, it is what eventually gave rise to aspirin. Aspirin and salicin are not the same thing, but aspirin is modeled on salicin. But again, the research that gave us aspirin arose out of this rather interesting finding about chewing the bark of the willow tree. In South America, natives use blow darts in order to bring down prey. And how does that work? Because the dart has a poison on it, and the poison is extracted from, it's actually a vine that grows on trees, which gives us curare. Curare was a huge breakthrough in medicine. And the reason for that is because it prevents muscle spasms during surgery. Surgeons don't like it when they make an incision and the muscle goes into spasm, and curare counters that. And this was a huge breakthrough in the 1940s. It made surgery much easier. Today, there are better drugs than curare, but curare was the drug that evolved from observations that the stuff that was on the tip of the blow darts had toxic properties, that it paralyzed the prey. So that gave rise to curare. Then sometimes drugs rise out of folkloric stories. For example, in ancient Chinese folklore, all kinds of herbs were used as remedies. Well, turns out that sometimes there is truth to these. For example, centuries ago, in fact, millennia ago, in China, malaria was treated with a tea made from the leaves of this plant. It's the Artemisia annua plant, and that contains a compound called artemisinin. And believe it or not, today, that is still one of the prime drugs that is used in the treatment of malaria. Malaria is a terrible disease. Generally, people think that it's a disease of a bygone era. This is not true. There are millions of people every year who die of malaria. I mean, not in Montreal, but in Africa and in Asia, where mosquitoes are rampant. It's a mosquito-borne disease, and malaria is a real scourge. And artemisinin is one of the drugs that is used today. And in fact, virtually every pharmaceutical company today has a branch that investigates folklore, investigates what medicine men, what shamans recommend to their people, because sometimes there is truth that comes out of it. I mean, most of the time not, but you never know. So it's always worth investigating these old folkloric stories. Sometimes medications can come from observing animals. That's a fascinating story. Chimpanzees, for example, will self-medicate, believe it or not. They seek out certain plants and chew on the leaves or the stems or the bark in order to alleviate problems. And observation of this has resulted in some real medications. It is written up in medical journals. This is one very interesting paper that discusses this whole business. And they observed a chimp who had a hand that had gone limp for some reason. In retrospect, it was probably some sort of arthritis. Anyway, the researchers observed that monkey seeking out a particular fern, turned out to be this one. And as they watched, he ate this every day, and they watched as his hand, or whatever you call the chimp's appendage, I guess it's close enough to being a hand, it normalized. So, of course, this meant that investigating the components of this plant was worthwhile, and they extracted a substance that is actually an anti-inflammatory, and it is now undergoing clinical trials, and we may eventually see this. So those monkeys, especially the chimps, have some amazing intelligence, often more than some people. Plants. I mean, obviously, we've referred to this in many examples already, but plants give us some very, very interesting medicines. You probably heard of Taxol. This is a drug, chemotherapeutic drug used for treatment of breast cancer, sometimes other cancers. And it is prepared by extracting the bark of a tree that grows mostly in the Western United States, also in Western Canada. Its chemical name is paclitaxel, and it's one of the most effective chemotherapeutic drugs. Of course, it took a lot of investigating before the exact substance was identified, because when you make an extract of any plant material, you'll get hundreds of different compounds in there. I mean, coffee, of course, is a classic example. You know how many compounds have been actually isolated from coffee? Over 1,000. People think it's just caffeine. No, there's a lot more in there than caffeine. There's over 1,000 compounds in coffee. So although we talk about deriving these drugs from plants, it takes a lot of work to do that. It's because you've got to separate all the components. But Taxol has turned out to be an extremely useful drug. And then we have colchicine. Colchicine is an extract of the autumn crocus, and it is still used today for the treatment of gout. And it has been used for centuries to treat gout. And again, this kind of arises out of folklore, because let's face it, in ancient times, if you had any kind of an ailment, what would you do? You try to eat or drink something. That was the only thing that you could think of. Now, most of the time, of course, these things didn't work and all fell by the wayside. But over the centuries, someone found something that worked. They told the story. Someone else tried it, etc., and became useful. Same thing goes for vincristine. This is extracted from a flower. It's called the Madagascar periwinkle. And it is today an effective cytotoxic agent. Cytotoxic means it kills cells, and the kind of cells that it kills are cancer cells. So this is a chemotherapeutic drug. Today, of course, we have many, many medications for hypertension, for high blood pressure. But the first one that was effective was Reserpine under a trade name Serpacil. Anyone remember that? Anyone having taken Serpacil? I mean, this would go back 50 years. I guess 50 years ago, you didn't worry about hypertension. Today, of course, we have a whole slew of drugs to treat high blood pressure. But this extract of Rauwolfia, which is also known as the snake root plant, gave us Serpacil, and there were many formulations of Serpacil. It was extremely popular because it worked. So we already see some mechanisms by which we, have insight into drugs. Today, of course, we have theories and evidence about how the body works. So you can develop drugs based on known biochemical mechanisms. For example, we know that Parkinson's disease is due to a deficiency of a chemical called dopamine in the body. So when that was discovered, then of course you start thinking, is there some way to replenish in the body the substance that is in low supply? Well, it turned out that you can't give dopamine directly to a patient because it is very quickly broken down in the body. It never gets into the brain. But a little modification of the dopamine molecule resulted in levodopa, and levodopa acts like dopamine, and it can cross the blood-brain barrier. So it gets into the brain. But that drug was developed based on knowing the mechanism of action of the disease, that Parkinson's is due to a dopamine deficiency. And today, of course, carbidopa, as is known, levodopa, is the classic treatment for Parkinson's disease. Then you can develop a new drug based on other drugs. When you find that one drug works, you try to improve upon it, usually by massaging the molecular structure a little bit and hoping for the best. A classic example there are Librium and Valium. Librium was introduced as an anti-anxiety drug in the 1950s, and it worked. But of course, as soon as you find a drug that works, you want to find one that works better. I mean, that's the natural sequence of events. And that gave birth to Valium. But this was because by the time that this drug was developed, we knew a lot about molecular structure. So that's the molecular structure of Librium. So if you want to improve upon it, what you do is make small changes in the molecular structure, hoping that you get a better drug. Most of the time you don't. Most of the time research doesn't work, right? But you never know a priori what will work and what will not. But even if you don't know anything about molecular structure, you can take a look at these two and you see that there's similarities. So Valium, very effective anti-anxiety agent, was arrived at by altering the substance of this molecule. Of course, there would have been many other alterations that never amounted to anything. But eventually they hit upon Valium. Then some drugs are discovered by accident. And the classic example there, of course, is Alexander Fleming and his discovery of what turned out to be the most famous antibiotic at all, of course, is penicillin. And Fleming was indeed looking for antibacterial substances. I mean, by the time that Fleming was doing his work, of course, Louis Pasteur had formulated the germ theory of disease. So it was known that bacteria and other microorganisms cause disease. So there was the question of what can you do to try to counter these. So what Fleming was doing was looking in a culture dish. He was growing some bacteria, and he was testing to see what kind of chemicals could kill the bacteria in the Petri dish, which is always the first step in research. I mean, you see whether or not something works in the lab, then you try it in animals, and finally in humans. Well, as the story goes, probably true, he went away for a weekend, and he left his Petri dish in the lab, sitting in the lab, and the window was open, and a mold flew in through the window and got into the Petri dish. And when he came back on Monday morning, he looked at it. He saw a mold. Chances are most people would have said, hell, this thing went moldy, I better start all over again. But he looked carefully, and what he noticed was that around the mold, the bacteria had died. So he figured that the mold was releasing some antibacterial substance. Fleming was never able to isolate that, even though he tried. And it took another 10 years until Florey and Chaim in England were able to isolate the active ingredient in that mold. And that, of course, was penicillin, which in a sense changed the outcome of the war, because the Allies had penicillin and the Germans did not. And the Germans suffered from all kinds of bacterial diseases that the Allies did not suffer from. So eventually Fleming, Florey, and Chaim got the Nobel Prize for discovery of penicillin. But it's a very interesting story, because Fleming's work was 10 years before the others. They never knew each other. But of course, it would never have happened if it wasn't for Fleming's original discovery. But the truth is that Fleming never managed to isolate penicillin. All right. So, as I said, all discovery begins with an idea. And once you have an idea, and we've seen where ideas can come from, it can come from animal observations, it can come from looking at plants, it can come from accidents, whatever. What do you do once you have an idea and you suspect that you might have in your hands a potential drug? The first thing is you have to test. There are two basic kinds of testing that are initially done. One we call in vitro, which literally means in glass. But what we're talking about are laboratory studies that are done in a test tube or in a Petri dish. Animal studies obviously use animals, mostly rodents, but an in vitro study. For example, what I just described to you, Fleming's observation of the penicillin was an in vitro study. I mean, it was an accidental finding, but it was an in vitro study. But today, for example, if you are searching for a new antibiotic, you would do this systematically. You would have bacteria growing in culture dishes. You would expose the bacteria to different substances and see what happens. And then, if you find that something has a potential use, you find some substance that is killing the bacteria in your Petri dish, what do? Well, then you take the next step. And this is where animal studies come in. And there are two things that you want to find out when you're using animals. One, very important, is the stuff safe? And second is, does it have a chance of working? Now, the safety is the one that is really important with animals, because, of course, humans are not giant rats. We have different diseases, so it's very difficult to study human diseases in rats. There are some, I mean, you can induce cancer in rats, for example, and then you can study chemotherapeutic drugs. But really, the first thing that you want to know about any substance that you think has potential to be a drug, is it safe, right? We're always looking at a risk-benefit ratio. I mean, of course, all drugs have some side effects, but you want the benefit to significantly outweigh the risk. So the first thing would be you do studies on rats, and you determine what is known as the LD50. not a pleasant thing to think about. This is the dose that kills 50% of a rat population. So let's say you start out with 100 rats, you start giving them whatever you're testing in increasing amounts, and when half of them have died, that's your LD50. So of course, what you want is an LD50 that is a very high dose, because that means that it's a pretty safe drug. There are a lot of animals that are used in research. It is estimated that every year just in North America, there are about 30 million rodents, mice and rats, that are used in research, about 200,000 dogs and cats and monkeys. And as you can, I'm sure, guess, there is opposition to this. PETA, People for the Ethical Treatment of Animals, want us to say no to animal testing because in their view, the life of an animal is as safe as the life of a human. To them, a fly is the same as a person. They don't even want a fly to be hurt. And they dig up these stories, such, for example, when they were doing some research on pomegranate juice, whether or not it could be used as a treatment or a preventative for heart disease. So PETA claims that mouse pups whose mothers were fed pomegranate juice in a chamber with almost no oxygen for 45 minutes in order to induce severe brain damage. Babies were decapitated and their brains were removed for the study. I mean, these are the kind of things that, PETA puts out. I mean, I don't know if this is true or not. I suspect not, or certainly not exactly this way. But the fact is that there's no way that pharmaceutical research can move ahead without using animals, because we can't use people. You can't take a substance that you think may work as a drug and have a group of people, give half of them the drug and give the other half a placebo, see what happens. I mean, no ethics committee would ever approve anything like that. But there's just no way that one can know the safety, efficacy of a medicine without doing some animal testing. I mean, one of the classic examples, of course, is the development and discovery of insulin by Banting and Best. I mean, this is one of Canada's greatest contributions to medicine. The 1921 discovery of insulin, which could be extracted from the pancreas of a dog, changed the life of diabetics around the world. I mean, before insulin, diabetics had, juvenile diabetics had a life expectancy of maybe 18 to 20 years. With insulin, they had the same longevity as the average person. I mean, this was just an absolutely stunning breakthrough. It would never have happened if it were not for using dogs to take a look at their pancreas and extract the juice from the pancreas. So nobody, of course, likes to think of animals being used in this way. But the question is, the risk-benefit ratio. Is it worthwhile to develop medicines for humans with some suffering to animals? I mean, it's not a simple answer to that question. I mean, a lot of people would say that it's not worth it, that the animals have the same rights as humans. Anyway, we use animals for sure, and PETA is sometimes, just a pain in the ass for researchers. There was one time, this goes now back about 40 years ago, when they came into our medical building, McIntyre building, and they, quote, liberated the monkeys. And they were running all over the place. I mean, it was terrible. Okay. So anyway, now we have some idea of where you get ideas in order to develop a drug. So the first thing that you do is an in vitro and an animal study. And if it turns out that you see some positive result there, then you could go to the next stage. But keep in mind that most drugs do not make it past that stage. That you do your in vitro experiments, you do your experiments with animals, but the results are not good enough to move ahead. If, however, you do find something that is safe and effective, then you go to the government and you apply for an investigational drug application. You have to describe what has been known so far in terms of your in vitro and your animal studies and what you are planning to do. And if this is granted, then you go ahead and you carry out what is referred to as a phase 1 study. Now, this is something that has to be reviewed by an institutional review board, and every university, every hospital has such a board where the researcher will describe what it is that they want to do, and the committee either approves it or not, taking into account how likely is it that a positive result will be found, and how unlikely is it that there will be some problem that happens. So anyway, if it turns out that the review board says, okay, go ahead, the first thing in a phase 1 study is to select some subjects, and these are a small number of subjects, as you see, usually 20 to 80, and you give the drug to these people. The only object of the study is safety. There's no question here of efficacy. You're not looking to cure any disease. You enlist volunteers who are willing to take that medication for a set period of time, usually for pay, right? And the only thing that you want to determine in a phase 1 study is whether or not the substance is safe enough to explore further. If it turns out that it is safe enough, then you can proceed to a phase 2 study, which is larger. You enlist hundreds of subjects, but this time the subjects are those who actually have the condition that you're trying to treat. So it's already harder, of course, to enlist several hundred subjects for that, because such a study will take several years. I mean, whether you're looking at a diabetes drug or you're looking at a cancer treatment or heart disease treatment, you know that these are not drugs that are going to be effective the next day or the next week. This is something that you have to examine over a long time. And not only do you want to have people with the disease, but you also want to have a control group, because you want to know whether or not what you're doing is effective. And this is why your control group is going to take a placebo. But nobody knows who is taking what, because the mind can play all kinds of tricks. So therefore, you have to have some knowledge of how your drug is going to work against a placebo. All right. So you organize this trial, which, as you can imagine, is not so easy to do. It's logistically difficult. It's economically difficult. But then if you're successful and you find that the medication is safe and it actually works, then you go to your phase 3 studies, where you have thousands of subjects. Much more difficult to do this, because you have to enlist all these subjects, and you will follow them for about 5 years. That's the accepted period of time. And eventually, most of the medications that have gone to phase 3 studies are approved. But as you can see, it takes a long time. So then, when you've done all this, then you go to the government with your new drug application, where you list everything that you've done, you list all of your results. There's a government committee. Here it's Health Canada. In the US, it's run by FDA. And they make a decision on whether or not they're going to have a formal review of this drug. That is the information that you submitted compelling enough to be looked at seriously by a group of experts? So they will make that decision in about 60 days. And then they will review the labeling that you want to put on the drug, the facilities where it is going to be manufactured. And after about a year, they make a decision on whether or not the drug is approved or whether or not it's going to be rejected. So as you can see, it's a pretty long process. And then even after a drug is approved and it's on the market, there's what is known as a phase 4 trial, where the people who are taking this drug are followed for up to 10 years. So it's a pretty good system. However, it is not foolproof. And a classic example of that is the thalidomide story, which of course many of you will remember because this goes back only to the 1960s. When a medication, thalidomide, Kevadon was the trade name, was given to women who were pregnant and were suffering from morning sickness, for which it actually worked. But unfortunately, as you know, it turned out that about 10,000 babies in the world were born with malformed limbs. It was a very tragic story. How did it happen? The truth is that it could probably still happen, because all the procedures that I just documented for you were properly followed. It just turns out that during pregnancy, if you take it in the second trimester, it can cause this problem. But the drug was tested in monkeys. It was even tested in women in the first trimester. It just turned out that it had this very, very bizarre effect that when it was taken in the second trimester, it caused this problem. As I said, there were 10,000 babies born in the world, malformed. Very few in Canada and very few in the United States. Most of them were in Europe, mostly in Germany, where the drug was first approved. And the reason that we didn't have this tragedy here was thanks to a McGill graduate, Dr. Frances Kelsey, who was given a presidential medal, as you can see, in 1962, because at FDA, she was in charge of approving the drug. And when she read all of the research, she was concerned that there actually had been some studies where thalidomide caused neurological problems. Some people complained of tingling in the fingers and toes if taking thalidomide. In Germany, when this was reviewed, they thought that that has nothing at all to do with morning sickness. But Dr. Kelsey was adamant, and she was concerned that maybe there's some neurological complication, which of course turned out to be correct. And because she never approved it, she kept asking for more and more data. It never made it to market. And eventually, of course, they found the babies, and then obviously it wasn't going to be marketed. But she was threatened by the pharmaceutical company because she put a stumbling block in their way. There are other drugs which have been approved, but only during phase 4, which I said you're going to follow them for about 10 years. Only then did problems crop up. Some of you may remember Hismanal. Hismanal was one of the first antihistamines that was put out that was not sedating. So it had a big market. But it turned out that it was associated in rare cases with fatal heart rhythm, and it was taken off the market. The same thing with Seldane, which was a very effective antihistamine. And then there was Lipobay, which was a statin drug. That was to lower cholesterol. It was manufactured by the Bayer company. It actually was effective in lowering cholesterol, but it turned out that it had to be withdrawn from the market because of muscle weakness and kidney failure, you can see. And then it was the weight-reducing drug fenfluramine, which made a big splash because it was very effective in terms of weight control. And it was approved in 96, and it was withdrawn very quickly in 97 because of the heart valve abnormalities that cropped up. Now, how do we explain this? Because remember that even in the phase 3 trials, you're only dealing with a couple of thousand people. So if you have a side effect that, let's say, affects 1 in 10,000 people, you're not going to find that in your phase 3 trials. But you will find it when the drug is out there being taken by millions of people, right? So you can never guarantee absolute safety, because if you have a very low incidence of a side effect, you're not going to find it in your clinical trials. Then, of course, there's the story of Vioxx, one of the most effective painkillers for arthritis ever introduced. This you must remember. Anyone remember taking Vioxx? By all accounts, it was an excellent drug, right? It took away pain, but it was withdrawn when some complications came up, heart complications. Again, it would not have been found in the phase 3 trials. However, there was another issue here. And the other issue was that supposedly Merck knew about these side effects from their own internal trials, and they hid that. And that, of course, becomes, sort of a criminal act. However, I think most people who took Vioxx right now would gladly sign a form saying that they will take all the risks if they could have it back, because it worked better than Celebrex, which was its replacement. There are all kinds of problems with clinical trials, to be sure. First of all, there are unforeseen risks. I mean, you start a human clinical trial based on your in vitro studies, based on your animal studies, but that does not mean that you're not taking a risk with humans, because, as we said, mice and rats are not people. So there's always the possibility, anytime someone signs up for a clinical trial, there's always a chance that something can go wrong. Then there are unnecessary trials, and this is a very big problem. Pharmaceutical companies, of course, are in it for the money. I mean, there's nothing secretive about that. There's nothing wrong with that. Every industry is in it for the money, okay? And because pharmaceutical products run on patents, which I'll describe in a minute, when that patent runs out, then anyone can sell the drug. So, of course, there's always the impetus to come up with a new substance so that you can patent it anew. So you may make a change in the molecular structure of a drug that you're selling. Why? So that it becomes a new drug that you can patent, even though it may not be any better than the one that you're selling. But it's different, it's new. So, of course, you have to go through the trials, but they may be unnecessary because your initial data shows that it's not better. It may be equal to, but not better than an existing drug. So there are these unnecessary trials. Then there is the problem of monitoring. People are difficult to monitor, right? I mean, for example, when you're doing a trial of a medication, and let's say they have to take the drug 3 times a day at a certain time with, let's say with food or without food, whatever. How do you know that they're actually doing that, right? I mean, these people are not living in a clinical facility. They're, out there in the world. And it's very difficult to know whether or not they are doing exactly what they should be doing. Then there's the problem that some people just may not understand exactly what it is that they are to do. Because often, the people who register for these trials are not the sharpest knives in the drawer, right? Because often they do it just because they're paid for it. And it's not clear whether they completely understand what they are to do, if they can follow the instructions. Now, there's another problem, which is financial conflict of interest. Researchers may have such a conflict. This study, University of Toronto, they analyzed 70 studies of a heart medication. Well, it turned out that 96% of the supportive researchers had ties to the pharmaceutical company that manufactured it, and only 37% of the critical researchers had drug company ties. This is a very big problem in research. Not only in terms of the papers that are published, which may be biased because of research grants. The bigger problem is papers that are not published because the results are not to the liking of the whoever is funding it. For example, let's say a researcher wants to study whether or not eating soup every day reduces the risk of hypertension or whatever. Okay? So where are you going to go to get funding for such a study? You're not going to go to a light bulb manufacturer, right? You go to Campbell Soup. All right. And then Campbell's is likely to come up with money for this because, of course, if the findings are according to their liking, they're going to promote it as a therapy, right? Soup therapy. But if the research shows that it is ineffective, it makes no difference, you may never publish that because you'll never get another cent from that company. So research that is not published can be a real problem. That's why today there's a big movement that any research has to first be filed with the government as a clinical trial so that anyone later can check what happened and whether or not the results were eventually published. Another interesting aspect of putting a drug on the market is how you're going to call it, the naming of drugs. And that becomes very interesting. For example, take a look at Viagra, which of course is one of the most popular drugs out there. Where does the name come from? Well, before a drug is put out on the market, there are all kinds of company committees that look at possible names because it makes a big difference. It has to be memorable for the person, and they have to be somehow able to associate it with the product. Viagra comes from vigor and Niagara Falls, because Niagara Falls is very potent, right? So you're talking about vigorous potency for Viagra. Cialis, which is a competitor for Viagra, comes from Ciel, which is the sky. The sky's the limit. Then you have Lipitor, which I'm sure many of you are taking. This comes from lipids, which are fats that you're trying to control, and organic, because organic has a good connotation. Xenical was a drug for weight loss and X out calories. So they come up with these names. I mean, it's not random. Ozempic, which is, of course, the most widely publicized drug today, the name has no meaning. There's no connection to anything. It was a committee that came up with this random word because they thought, upon studying it, that people would remember it, and we have it. But it wasn't based on anything. Now, as you probably know, in the United States, you can advertise pharmaceutical drugs, prescription drugs on TV. And If you watch the nightly news, whether it's CBS, NBC, or ABC, every commercial is for a drug. Why? Because it's old people like us who watch the news. I can tell you that my 21-year-old students do not watch the news. Whatever information they get is from TikTok or Instagram, and if it's not on their cell phone, they don't know it. Okay, but of course, people of our age watch TV, so they will advertise in the U.S. For example, the anti-diabetic effect of Ozempic. It lowers your A1C. They can advertise the fact that it leads to weight loss. In Canada, you cannot do that. You cannot advertise a drug saying what it is for. So how do they get around this? With some very clever advertising, which of course you've seen. And we just ask, go and ask your doctor. So just throw out the name, Ozempic, go and ask your doctor. You cannot say what it is, what you should be asking your doctor about, what it's for, but it works because people remember the name, they'll ask the doctor. There's a very interesting and important issue to consider here with Ozempic. There's a lot of fake Ozempic on the market. Ozempic is very expensive. It's about $1,000 a month, right? Which of course means that people are going to, get into the fake market. And fake means that there's no active ingredient in there. It looks the same. It's the same syringe. It looks identical, but there's just nothing in there. It's a total fake. And this happens when you go on the internet and you try to buy. You cannot buy legitimate Ozempic on the internet. It's a prescription drug. You cannot get it online. There's another aspect here, is that there's something called a compounding pharmacist. And supposedly, they can make up semaglutide, which is the active ingredient in Ozempic. To tell you the truth, I don't know what these compounding pharmacists do, but they cannot be making Ozempic. It is a very difficult substance to make. I mean, there is actually a reason for the expense for the $1,000 a month. It's very complicated to make this drug. I don't know what these compounding pharmacies are doing. So of course I wouldn't trust any Ozempic that they sell. Now, interestingly enough, in Canada, we are soon going to have generic Ozempic. You know why? Because once you get a patent, you have to pay a yearly upkeep for the patent to be active. Believe it or not, it's hard to believe. Novo Nordisk, which is a large Danish pharmaceutical company, the one who came up with Ozempic, the guy who was in charge of having to pay the annual fee, which is a minimal annual fee, to renew the patent, for some reason did not do it. And the other pharmaceutical companies got wind of that. And it turns out that the patent is no longer going to be effective in Canada. So anyone can manufacture Ozempic and sell it. Of course, it doesn't mean that it's going to be cheap, because it is still, if they're going to make the real stuff, it's still difficult to make. But when many companies can get into this, the price will indeed come down. Now, with names, there are also naming problems. For example, some drugs have very similar names. Celexa, Cerebrix, Celebrex sound the same, but they're very different. Now, this used to be a significant problem when doctors hand wrote prescriptions, because the pharmacist could easily make a mistake. Today, this is not such a big problem, because prescriptions are not handwritten. Everything is computerized. Now, of course, advertising. There's obviously a great deal of money spent on advertising drugs, because it's a huge business. 12 billion a year, and about 1/10 of that in Canada. That's the usual ratio. Just advertising medications. That's a lot of money. They actually spend more money on promotion of their drugs than on research and development. And they spend more money than consumer products like, beer and cosmetics. Pharmaceuticals spend more on promotion. Now, the pharmaceutical business has what we call patent protection. That is, when your drug has been approved by Health Canada or FDA, you can sell it. No one else can sell it because it is under a patent that you have filed. And the patent protection is 20 years. So for 20 years, from the moment that you file your patent, only you can sell that drug, which sounds far better than what it really is. Because a patent is filed the moment that you start researching a medication. And it generally takes 10 to 15 years to get the medication to the market. So the fact is that you're only getting maybe about 6 years of patent protection. Now, that, of course, is still significant, but patent protection is what allows pharmaceutical companies to make money. And this is what allows them to do the research to come up with new drugs. So while, you can sort of try to denigrate this and saying that, why should they be given this license to, to make up their price and all of this? Well, because that's what allows them to do the research. Now, I think very often the pharmaceutical companies overstate that case, and they don't spend as much on research as, they lead people to believe. Once it's off patent, then a drug becomes a generic, which means that any company can sell it. And the question comes up, is it the same? Well, yes, but not 100%. You have to show that the generic works just like the product that it is copying. No question about that. It has to have the same bioavailability, etc. However, it does not have to have the same excipients. These are the other ingredients in a pill. When you make a pill, it's not only the active ingredient, because you have to have the filler in there, you have to have a coating on it. Sometimes the coating dissolves less or more easily. So it may not be 100% equivalent. And there are people who have found that when they take the generic version of a drug, it doesn't work exactly the same because it may not dissolve exactly the same, even though the active ingredient is the same. Now, the active ingredient, of course, often has a terrible chemical name, as you can see here. This complex name is for tetracycline, which is an antibiotic. Now, this antibiotic now is available generically because it's an old antibiotic. So it can be sold by any company, and it is. Every one of those is exactly the same thing. It's tetracycline sold by different companies, but the prices are not the same. As you can see, the differences can be absolutely dramatic. Going from Apotetra, which is the cheapest version of it, to Bristol's Tetrex. Or look at ampicillin, widely used antibiotic, often in children. Look at the prices. Exactly the same thing. Another one, diazepam, is Valium. Look at that. Huge price difference. Now, in Quebec, as you know, when you get a prescription, the pharmacist has to fill it with the cheapest available version, which is usually the generic version, except if the doctor writes deliver as written. So if the doctor writes Viagra, deliver as written, they have to give you the Pfizer version that is called Viagra. But if not, then they give you a generic version. Now, there's also a big difference between Canada and the US, which is why many Americans come here. The differences, as you can see, can be very significant. And of course, in the US, they're trying to do something about it to try to bring down drug prices. But I mean, obviously, the pharmaceutical companies and their supporters, which generally is the right wing in the US, of course, argue against that. So anyway, I think that all of this has given you an idea of where medications come from. It's such an interesting story. And all the steps that have to go through before you get to pop that pill into your body. And most of the time, you can trust what you're doing, but there are no guarantees, because there is no medication that doesn't have a side effect. And also, medications don't work the same way in everyone. We are biochemically individual. Yeah. And I can give you a very current example. I'm dealing with this stupid sciatica, and so I'm taking Advil. And when you look at the literature, it will tell you that Advil kicks into action after about 30, 40 minutes. I don't find that at all. I find it takes 2 hours until it becomes effective, but then it is effective for about 4, 5 hours. But someone else may take the same drug and they find that it does go to work in about 30 minutes. It all depends, on your enzymes, on how quickly they break down the drug or don't break down the drug. So there's a lot of trial and error with medications. Anyway, that's the story. So if you have any questions, yeah. Oh, I know all the stretching, believe me. Sorry, why they don't? Oh, well, I mean, it didn't happen overnight. Yeah. I mean, since the late 1800s, there was research into what causes diabetes. And the clue came when they tried tying off different organs in an animal to see what the effect would be. And when they tied off the ducts from the pancreas, it resulted in diabetes. So that was the clue. But there's many years of research there. I mean, I always tell my students, you look at your textbook, and there'll be a sentence in there about the drug and what it's for. Well, that sentence has probably dozens of years and thousands of researchers working on it before that sentence makes it into a textbook. Yeah. Yeah. Right. Right. Yes. Yeah. Yeah, I mean, there may be something to that. I mean, there's also a question of why we have the highest rate. The suggestion has been that it's because of vitamin D shortage, because we don't get enough sun exposure. Although I don't think that there are too many researchers in that camp who agree with that. So we really don't know why Canada has such a high rate. But uh, the approval process is a very long and arduous one. And in Canada, they ask for a lot of proof. And often what companies submit is what they call preliminary studies. And it often doesn't amount to anything. So, I mean, I'm not sure about that MS drug, but I mean, there are numerous MS drugs in the trial today. You know, if you look at clinical.gov, which is a website in the US where you have to register whatever study, dozens and dozens of studies on MS, all kinds of drugs that supposedly stop it or reverse it. So I don't know what that specific one is, but it's complicated business. Yeah. Uh, yeah, I'm not sure what the legalistics on that are. Yeah. Hmm. Yeah, I mean, there's all kinds of legalistics there about prescription. Yeah, because I guess the law is that they can't. I mean, it's yeah, yeah, yeah. Well, the U.S., as we know, is a quagmire now in many ways. Yeah, yeah. The what? Placebo effect, the usual statistic that is quoted is 30 to 40%. Of course, it depends on what kind of condition. I mean, the one that is most susceptible to placebo is pain. So you're talking 30 to 40%. Yeah. Yeah. Yeah, I did. That was last night. Yeah, I didn't see it, but I assume it was good. Yeah, I mean, Sanjay is very reputable. Yeah. So uh, I suspect he was going on about how there is some medication for all pain and that there's no need to suffer. Yeah. Meditation. Yeah. Well, uh, I'll trust the Advil. Yeah. Yeah. And what? No. Sure. Sure. I mean, Thorazine is one of the most famous psychiatric drugs. It basically emptied sanitariums of people who had schizophrenia because it was a very effective treatment for schizophrenia. Yes, yes. I mean that's true. Yeah, but that's a prime example of a benefit risk analysis, because schizophrenia is notoriously difficult to treat, and Thorazine was the first really effective treatment. And yes, it did. It what? Yeah, Thorazine is chlorpromazine. Yeah, yeah. It's called what? No. But anyway, I mean, yes, I mean, there's a heart disease complication, but when you have schizophrenia, you take that chance. I mean, you have to look at the percentage of complications. One thing that, you should be aware of is when you watch those ads on TV every night about the drugs, and of course they list all the side effects, and then, you always say, why the hell would anyone ever take this medication, right? But the law is that side effects have to be listed even if there's only one documented case of that happening. So the fact is that the side effects occur very small percentage of the time, because if it wasn't a small percentage, the drug would not be approved. But of course, you can have side effects. I mean, as I discussed. All right, so see you next month.